welcome everybody. Good evening, and we'll continue with our discussion regarding with the course Basics of Neurobiology, right? So just give me a moment. I will share my screen. Yeah. Okay. So welcome everybody. As we have already, let us begin with the introduction over here, and then I'll do a quick recap of what we have already studied in the last discussion session, right? So we have been going through with this course that is Basics of Neurobiology at this platform ISSS. And in this course, up till now, what we have done is we have learned about like what are the basic, what is the history behind this field, neuroscience, neurobiology, who were the major scientists that were involved in this field, what is the current understanding of the brain and the nervous system that we have, how did we reach to this understanding, what is the general morphology of the brain, what are the types that are there in the nervous system, what are the kinds of nervous system that are there. Then, along with that, we have also studied some functional perspectives of the brain. And then we have gone through what are the different techniques that are used to study the brain, right? And even stimulate the brain at that. Then we had begun with the discussion upon brain development and how that happens, what is the molecular level mechanism behind that. And we are going to continue with that discussion, and we are going to finish that discussion in today's lecture. Along with that, what we will also understand, which is also part of brain development actually, is how the brain is also sexually dimorphic, right? So, what is sexual dimorphism? We will discuss when we come to that part of the discussion, right? So, this is lecture number 7 for this particular course, Basics of Neurobiology. It is the second last session, I think. We will wrap up this course. Maybe we will not go into the depths of how memory is working, right? Maybe we can have another, course or, sub part of this course later on. But for now, on Sunday, we will have the final session for this particular course, Basics of Neurobiology. Before we, move forward as to understanding the development of the brain, let us quickly give a recap as to what we have already studied, right? Because this is by far been the most terminology induced kind of session, right? So there were a lot of new terminologies that were introduced to you guys. Then like it might have sounded like jargons, but the names are such, or the signaling molecules, transcription factors, they have certain names, and we cannot really simplify it much further if we want to study the details. But for people who are not from a biological background and who only want to understand the overall scenario regarding how the brain is developing, my suggestion is that you do not go into the details of the naming or remembering the names of specific things. You just understand the mechanism, right? Understand what is going on at that time, right? So since it was a very packed session last time, we will do a quick recap of that session. I am unable to switch like this, so maybe yeah. So I have kept the slide open from where we actually delved into the, depths of developmental biology last time with respect to nervous system. And over here, you can see we first talked about the patterning of nervous system, right? What does patterning of nervous system mean? It basically means that how the diversity of neuronal cell types that are present in the mammalian nervous system, how are they differentiated or how are they distributed across the nervous system, right? So first we saw that neural tube is actually the first nervous system component that comes into existence, right? And prior to this, there is a primitive nervous system component that is produced, which is basically known as a neural plate, right? So that neural plate is arising from the ectoderm, right? So at this stage of the embryo, the embryo is already differentiated into three major, segments: ectoderm, mesoderm, and endoderm. We discussed them last time. And the one that is responsible for nervous system development is ectoderm, right? So what happens, how does nervous system develop from this is that in the ectoderm there are some region in which basically develops as a neural plate. Right now, what is this neural plate? This neural plate is the region from which the entire nervous system thereafter will be formed, right? So during development there is this region that is present, which is neural plate. Eventually, during development, this neural plate, it forms a ridge, and this ridge basically grows further. It forms a groove-like structure. It is known as the neural groove. This groove, further during development, what happens is the corners meet and it becomes like a tube-shaped structure like this. Now it is known as a neural tube because the end has joined over here, right? So then this neural tube structure, it will form an actual tube, and the mesoderm beneath it will also develop and it will form something that is known as a notochord, right? So this is what the final primitive structure still of the nervous system looks like, right? Yeah. Uh, then we saw that the brain that is formed has also is sequentially formed. It is not like the entire structure of the brain is always there, right? So in that neural tube itself, there are different stages during embryonic development that are present. The first stage is a three vesicle stage, right? In this three vesicle stage, you can see there are three major vesicles. They are the proencephalon, mesencephalon, and rhombencephalon that are present. Later, when the brain is finally fully developed, we have a four vesicle structure, right? We have studied it before when we were studying the gross organization of the brain, right? Uh, four major vesicles over here we will list. There are different kinds of naming for that. Over here it is listed as a five vesicle stage because they are also still counting this myelencephalon as one of the vesicles, right? Over here, over there, while studying of gross brain development, we had basically studied diencephalon, forebrain, midbrain, hindbrain. So four in that sense, right? So these are the structures that are formed. You can see that proencephalon has basically divided into two, 1A and 1B. 2 has remained constant. 3 has again divided into two, 3A and 3B, metencephalon and myelencephalon, right? So that is how differentiation of the nervous system is continually happening as and when the embryo is developing, right? So, these sequential stages of neural development are as follows. Over here, you see that the embryo, you might know, is this is the fetal position in which the embryo is present, right? So, inside the mother's womb, this is how the embryo is generally present. So, as this embryonic development takes place, this is the three vesicle stage, 1, 2, and 3. As it develops further, and this 1A, 1B, 3A, 3B is added, you can see that some flexures or some points wherein the bends occur are also further added, right? Three major flexures that we talked about was cephalic, pontine, and cervical flexure, right? Then we also saw that there were secreted signals which promote neural cell fate, right? Why did we think like that? Because we basically knew that there were two major classes of proteins that work together and promote differentiation of ectodermal cell into a neurodermal cell, right? So either it could be an inductive factor or the second could be the surface receptors which are receiving that factor and then acting upon it, right? So that is what we thought, that there is something that is going to induce neural development or neural cell formation, right? As in when we went through this particular course, we finally found uh, that indeed there is a specific region that is present in the embryo, which is basically known as the dorsal lip of the blastopore. It was found by these scientists, Mangold and Hans Spearman, and it is known as the Spearman-Mangold organizer, right? And they found basically that this dorsal lip of blastopore is essential for neural fate development, right? So what they did was this: if this region is present, it is the organizer. It is not forming the neural nervous system by itself, but it is inducing the surrounding cells to form the nervous system, right? So what they saw was that if this was present, then it induced formation of the neural tube above it, right? Which will eventually lead to the nervous system formation. But they if they basically transplanted another one of this dorsal lip of blastopore from another donor embryo into the same embryo, they saw that there were two neural tubes that were actually formed, right? So this was inducing the surrounding region basically to form a neural tube, right? So that is why it was known as the organizer region, because it is organizing the cells nearby to it to basically develop into a neural fate, right? But at this point also we knew that okay, there is this region that is responsible. We did not really know about the molecular details that went behind this kind of patterning, right? So studies went by, right? And we finally found that the default fate of ectodermal cell is actually neural differentiation, right? Upon multiple other studies, it was found that it a neural cell formation doesn't have to be induced. It actually has to be repressed, right? Which means that by default, if a normal cell is present, a normal ectodermal cell, if it is isolated, it will always, in isolation, it will always by default form a nerve cell, right? Only if there are some factors that are still present or multiple clusters of cells that are present that will signal one another some kind of factors, will they develop into skin cells or epidermal cells, right? Individual ectodermal cells will always by default form nerve cells, right? So that was what was found, right? And this was very, very characteristic. We Nobody thought in that sense. Everybody was looking for an inducer for neural cell formation. Now it suggested that instead of an inducer, there was actually a repressor that was present, right? Because the natural fate is neural fate, and that is being repressed in some cells to form the epidermal fate or the skin, right? So, this was basically then studied, and it was found that neural induction is mediated by peptide growth factors and their inhibitors, right? So, these growth factors, such as FGF, right, or inhibitors such as BMP, etc., right? So, there were certain proteins, such as bone morphogenic proteins or BMP, right, and TGF beta related proteins these factors and proteins, right? Now, what we studied was that this BMP is actually important because whenever this BMP signaling is present, the cell will have an epidermal fate, right? But if the organizer region is present close by, it will basically have inhibitors of BMP that are present, so that the region close to that dorsal lip of blastopore will because these BMP inhibitors are activated, BMP signaling will not be active, and those cells will be able to form neural cells, right? But So, if BMP signaling is present, it will promote epidermal fate. And if BMP signaling is disrupted due to inhibitors such as cordon, noggin, follistatin, then neural fate, which was the actual default fate, will be taken. Right? So, that is what was seen. Also, it was studied that if we dissect basically this region of ectoderm, what we see is by default, it will form epidermal fate if it is present as a cluster. If this cluster had BMP inhibitors such as these three, one of these three also given to it, it will always form neural fate. If these cells were dissociated, single cell suspensions were made, then also it will form neural inhibitors. But if it was dissociated and BMP was added along with that, it will again revert to epidermal fate, right? So, from this, what we understood was that ectoderm is responsible for nervous system formation. Ectodermal cells by default have the fate to form neural cells. The BMP signaling is basically inhibiting the formation of neural cells, and there are some BMP inhibitors that are present in the areas wherein BMP signaling is not activated, so that actually nervous tissue can be formed, right? Then we studied about the rostrocaudal patterning of the neural tube, right? So what is rostrocaudal? I told you rostro means always directed towards the nose and caudal directed towards the tail, right? So nose and back, front and back, right? So we saw that there is some kind of patterning gradient, that is, there are some kind of signals that will help in this patterning also. The major signaling pathway was wind signaling, right? So if this is a neural plate, right, we what we see is that there is some region wherein wind signaling is active, and there is some other region wherein wind inhibitors are present, so that wind signaling is not active, right? So wherever wind signaling is active, it will form the caudal region, the backside tail side region of the brain, and wherever wind inhibitors are present or wind signaling is inactive, it will basically form the rostral region of the brain, right? So forebrain and midbrain. And how will this be decided? Because this wind signaling or lack of wind signaling will activate different transcription factors, right? So in the forebrain midbrain area, OTX2 is the transcription factor that is activated. In hindbrain region, GBX2 is the transcription factor activated in the presence of wind signaling. And in between, there is a midbrain hindbrain boundary wherein a third transcription factor engrailed is activated, right? Which will have another distinct characteristic inside the brain, right? So that is what we studied, that along with this kind of wind signaling, there are also signals from the mesoderm or other developmental layers, right? So that was still a part of the ectoderm, right? But just below this, we saw that below the neural tube, we had seen prior to this, was a notochord that was present. Notochord is mesodermal in origin, right? So there are regions or signals from mesoderm such as notochord and endoderm such as precaudal plate, which will also affect the rostrocaudal patterning of the neural plate, right? So depending on what kind of signal this developing brain is getting from either the mesoderm or precaudal plate, they will further differentiate accordingly, and there will be differential expression of the brain or differential patterning of the brain that is done. Then I think this is the region where we left off, the dorsoventral patterning of the neural tube, which involves similar mechanisms to the rostrocaudal levels, right? So till the rostrocaudal patterning, actually, we had discussed in very high details last time. So that is why I wanted to quickly go through it and revise it also, right? Now we will switch to this dorsoventral patterning from this class onwards, right? So, so that we will go slower over here, that we were just revising, this we now have to understand. Okay. So, I have tried to cut down on, the very minute details or details that were not relevant for this particular topic, at least for this particular kind of lecture. At least if we were having a different kind of class wherein we were just studying the developmental basics of nervous tissue organization, then maybe I would have dealt with each and everything. Over here, I will try to touch everything, give you a brief idea about everything, but still not go in over or excessive detail, right? Okay. So let us now look at the dorsoventral patterning of the neural tubes, which involves similar mechanisms at different rostrocaudal levels, right? What does this particular line mean first, right? So what was rostrocaudal? It was facing the nose and facing the back or the tail, right? What is dorsoventral? Dorsoventral is the top of the head facing the sun, the bottom of the head facing the ground, right? That is dorsoventral. So what we will study over here is that even across this rostrocaudal axis, even at one locus, if you see, so if rostrocaudal will have forebrain in the front, hindbrain at the back, right? Even if I just pick up the forebrain and look at it, the top part of the forebrain might have a different kind of patterning, and lower half of the forebrain will have a different kind of patterning, right? That is a dorsoventral axis, right? Which is what we are going to study over here. So, as soon as the neural epithelium acquires its rostrocaudal character, so that is the first characteristic to develop, right? The rostrocaudal axis is the first one to develop in a developing, nervous system. As soon as that is done, the cells located at different positions across its dorsoventral axis start to differentiate into various neuronal and glial cell types, right? So the neurons in the spinal cord serve two major functions, right? So over here we will understand with the example of spinal cord neurons, right? So it is towards the hind end or towards the caudal end of the brain, right? So, what do these spinal cord neurons do? They will do one of the two major functions. They will either relay cutaneous sensory input to higher centers in the brain, and they transform sensory input to motor output, right? So, either they will take sensory input to the brain or they will bring motor output from the brain, right? That is the two major types of things that these uh, you know, uh, neurons, the spinal cord neurons will do, right? Then the circuits involved in processing of cutaneous sensory information are located in the dorsal half of the spinal cord, right? So whatever the sensory input receiving neurons, they are generally located in the dorsal half of the spinal cord, and those that are involved in motor output are located in the ventral half of the spinal cord, right? So this is interesting now. Even if you look at the spinal cord region, the dorsal half or the top half will basically have the region wherein sensory neurons are present, right? Cell bodies of sensory neurons might lie, right? And other parts also of those neurons. And the ventral half or the lower half is the one wherein motor neurons will lie, right? So the dorsoventral patterning of the neural tube is initiated by signals from mesodermal and ectodermal cells that lie close to the ventral and dorsal poles of the neural tube, right? And is perpetuated by signals from two midline neural organizing centers. We will soon look at that also, right? So this is it. Uh, there is this midbrain hindbrain boundary that is present, which is represented in this purple color. The signals from this midbrain hindbrain boundary basically pattern neurons in the midbrain as well as in the hindbrain, right? So there is some kind of FGF signaling over here that is being released from this isthmic organizer region, right? This FGF signaling, if it is going towards the midbrain, there is a higher level of sonic hedgehog signaling towards the ventral side, right? This higher level of sonic hedgehog signaling towards the ventral side will basically determine motor neuron fate of these particular neurons, right? Then again, in the hindbrain region, since along with this sonic hedgehog signaling, the notochord is also giving other mesodermal signals, and GPX2 also is expressed. So this combination will lead to formation of serotonergic neurons over here. Whereas over here, the transcription factor that is expressing is OTX2. Sonic hedgehog signaling is there, right? So this combination will lead to formation of dopaminergic neurons over here, right? This signaling combination. FGF is obviously present because it is affecting these regions from this midbrain hindbrain boundary, right? So, the rostrocaudal position is actually a major determinant of motor neuron subtype, right? That is what we saw, that whether it was going to become a dopaminergic neuron or serotonergic neuron, it was determined also by the rostrocaudal axis, not just by the dorsoventral axis, right? Dorsoventral axis, what did that determine? It determined that the ventral side will actually have the motor neuron, right? What is the subtype of that motor neuron is determined by the rostrocaudal position of this motor neuron, right? So motor neurons are generated along much of the rostrocaudal axis of the neural tube from the midbrain to the spinal cord, right? So we have a plethora of these motor neurons in the ventral axis, ventral side of this rostrocaudal axis, right? This suggests that one of the goal of the patterning signals that establish rostrocaudal positional identity within the neural tube is to make motor neurons different. So that is one of the role of this rostrocaudal, uh, you know, role that it is playing in the dorsoventral patterning also, right? One of the major class of genes involved in specifying motor neuron subtypes is the Hox gene family, right? So now this is very important because even when you ever come across any development course particularly, you will see that this Hox gene family is actually a very important gene family, right? It is conserved across a lot of different species also, right? So it is, we'll study more about this Hox gene family, but we will not go into detail about each and every Hox gene, right? We will maybe see an overall picture of what this Hox gene family is doing. These homeobox genes, Hox is actually small or short for homeobox, right? So these homeobox genes encode a family of transcription factors that contain a homeodomain, right? This homeodomain is actually a domain that is conserved across species, right? These homeodomain proteins represent a major a class of transcription factors that regulate developmental processes in organisms as diverse as yeast, plants, and mammals. Right? So again, the same thing. It is basically conserved across a plethora of species. Right? So this antero-posterior profile of Hox gene expression determines the subtype of motor neuron in the hindbrain and the spinal cord, right? So what it is saying is that which Hox gene is present across this anteroposterior profile, that is towards the top to the bottom. What is the Hox gene that is actually expressing will determine which is the subtype of motor neuron that will be present either when we are talking about it in the brainstem, that is a hindbrain region, or later down in the spinal cord, right? So over here you can see that we consider this part as the hindbrain region and this part as the spinal cord. So this is towards the top, so it is anterior. This is towards the bottom, so it is posterior. Right towards the anterior, you can see the Hox genes are that are expressing are Hox A2, Hox B2, Hox B1, Hox A1, Hox B3, Hox A3, and so on. Right till Hox C4. right? And each one of them is responsible for different kind of motor neurons that are going and basically innervating either the face, cornea, mouth, face, parotid glands, soft palate, and pharynx, right? So regions that are restricted to our head and our neck, right? So cornea is in the eye, parotid gland is in your cheek, and the soft palate is the upper part of your mouth, and pharynx is your this uh, the neck region, right? Just above esophagus is that pharynx. So these are the regions that are being affected by or that are being looked at by this Hox gene family that is present towards the axis of this anteroposterior posterior profiling of this hindbrain and spinal cord, right? Once we look at this spinal cord region, right, we see that there are different Hox genes that are expressing over there. Hox A6, Hox C6, Hox A9, Hox C9, Hox A10, and Hox D10, right? So different kind of Hox subtypes are over here, and they are basically leading to determination of different kinds of motor neurons that are going and that are innervating either the forelimbs or the sympathetic neurons or the hind limbs, etc. Right? Yeah. So let us look at the organization of this Hox genes a little more, right? So the clustered organization of Hox genes is conserved from flies to vertebrates, right? So flies were invertebrate animals, right? They do not really have a spinal cord, but and vertebrates are the ones that do have a spinal cord, right? Over here, you can see that the overall pattern of expression is similar, right? So the pattern has been represented by colors, right? So this means that the homologous uh, uh, Hox genes, they have been color coded over here, right? So generally, ABDB that is present in this Drosophila is similar to A9, B9, C9, D9 that is present in the mouse, right? So that is why they are given as similar colors, right? Zen is more similar to A3, B3, D3 that are present in mouse, which is why they have been giving similar codes, right? So you can see that these Hox genes that are present and that are conserved across species, you can look at the pattern of arrangement, right? You can actually appreciate the pattern of arrangement of these Hox genes, right? So over here, in case of Drosophila, you don't really have to get into the details of the names of this, but this antennapedia complex is the one that is going to be responsible for formation of the antenna and the head of this particular invertebrate, that is Drosophila, and these other bithorax complex is actually going to be responsible for the abdomen. Part of thorax was in this antennapedia complex, and part of thorax is in this particular complex along with the abdomen, right? Similarly, you can see that even in vertebrates, right? So they are several species apart when we talk about in the evolution lineage, right? But if you look at them, you can appreciate that the genes that were or the Hox genes that were basically homologous to these lab gene are still expressed in the anterior side of this fetus, and the genes that are more close in relation with this abdominal B gene are expressed in this caudal region or the posterior region of this particular fetus, right? So that that is how this Hox gene has a clustered kind of organization, which is basically conserved across species, right? Again, uh, as we had seen a couple slides earlier, that there are different kind of Hox gene subtypes that are present across different levels of, this anterior-posterior axis, and which Hox gene is present at what position is actually determining what kind of motor neuron is being found at that region, right? So over here we will understand that there are some local signals and transcriptional circuits that are present which further diversify the neuron subtypes, right? So we have already seen that if a combination of some transcription factors and growth factors are present, they can determine, and where they are lying in the anterior-posterior axis, they can determine what is a subtype of this motor neuron that is going to be formed, whether it is dopaminergic or serotonergic or cholinergic, what is the kind of neuron that it is going to form. Right over here, we will see how where they are present is also going to differentiate that, right? So over here you are seeing a wild type and a Hox B1 mutant, right? So Hox B1 mutant is a homeobox B1 mutant, right? So this means that this homeobox B1 or Hox B1 gene is probably not normal in this particular mutant, right? In this particular mutant, everything is normal. So over here in this normal type of animal, what we see is that this R2 is one region that is anterior to this R4 region that is present. This R2 region has a group of neurons that are coming out, or axons that are coming out from an area in this R2 region, right? And these are basically going and forming the trigeminal motor neurons, right? Posterior to that is this R4 region, which has another set of neurons that are sprouting out of this R4 region, and these are actually going and forming the facial motor neurons. Right now, there are different factors at play due to which there is difference in which kind of neuron is actually being formed. Right, over here say that Hox A1 is active, and over here Hox B1 is active, right? Over here we know in this Hox B1 mutant, Hox B1 is not going to be active as its normal self, right? So let's say over here in R2 again, Hox A1 is active, and over here where Hox B2 should have been active, it is not there, right? So because it is not there, this R4 will also behave like R2, right? So it will go back to its default fate, or the fate of its nearest neighbor, right? So that is why instead of these kinds of neurons that had a different fate, that were going to go and innervate the facial muscles, right? Now it will still form the trigeminal like motor neurons, right? Because it has changed in fate. Its fate has been altered because of absence of this HOXB1 gene, right? So not exactly trigeminal motor neurons, but similar to that, because it is in a close vicinity to this area, this kind of motor neurons will now be formed, right? So this basically tells us that local signaling through this Hox genes is also very important for the proper patterning of this motor neurons, and for proper differentiation according to which part of the body they want to go and innervate, right? So this is another summarization of the similar principle. Over here, you can see that the Hox protein control the identity of neurons in the motor columns and pools, right? So this is a spinal cord. It is a region of the spinal cord. This is the rostral region, and this is the caudal region. So this is closer to the head, and this is closer to the tail, or the bottom of our body, right? So from in between, this spinal cord has been cut, and that is represented over here. And you can see that there are these three sections in spinal cord that have been isolated, right? Now, these three sections, they have different kinds of Hox genes that are activated over here, right? That are acting over here. So this section one that is represented in this lavender color has HOX6 gene, and it is responsible for this later lateral motor column, right? So this LMC is lateral motor column, which is associated with HOX6. The one that is associated with Hox 10, this one is lumbar column, right? This is lumbar column. The one that is associated with Hox 9 is the preganglionic column, right? This one. So these are basically different regions of the spinal cord from which or at which different kind of motor neurons are present, which are going and innervating different parts of the body, right? So over here you can see, yeah. So over here you can see that uh, yeah, they are color coded also, right? So even in this LMC region, again, there is a differentiation between what is the kind of Hox genes that are active, and based on that, which are the subtypes or which are the transcription factors that it is further going to activate, right? So this is just a representation to tell you that different kind of Hox gene or gene pools, they will determine the fate of that region, right? What kind of activity that region will have, right? That is why it is so important in development, this homeodomain proteins, right? So over here in the second half, what you can see is there is this one structure that is wild type again, which is going to be the normal kind of region, right? So this wild type spinal cord section, you can see that there are two sets of motor neuron cell bodies that are present over here, and these two sets of motor neuron bodies have neurons sprouting from it and going and basically either innervating the FCU region, which is the flexor carpi ulnaris region, or the pec region, which is a pectoralis muscle region, right? But in case the RNAi is given for HOXC6, right? RNAi means RNA interference, wherein this HOXC6 gene will be inactivated, right? What will happen? This pectoralis muscle forming innervating neurons are now no longer present. Only this subtype is present, which will go and innervate only the FCU muscle, and thus you will probably not have any sensation or any response in this pectoralis muscle. So it will be almost not developed at all, right? If there was another kind of vector that was given for this HOXC6, wherein there is this megalovirus that is given with this HOXC6, due to which there is overexpression of this particular homeobox gene, right? Now, what will this overexpression lead? It will not let this other green one also perform its normal function. What it will do is it will pattern the entire region that was dedicated for motor neurons into motor neurons that are actually being differentiated by this HOXC6 intervention, and they are all going and now innervating the pectoralis muscle, and the FCU region is left as it is, right? So this tells us that there has to be a very careful balance between the levels of this Hox signaling also that needs to be present, right? Okay. So, the developing forebrain is also patterned by intrinsic and extrinsic influences, right? Over there, we have ended our discussion about the dorsoventral axis and the anterior-posterior axis also now. Now, we will talk about the development of the forebrain, right? This is a very brief topic that we are going to touch. Over here, you know what the forebrain is, right? The cerebrum region, right? So neurons in the mammalian forebrain form circuits that mediate emotional behaviors, perceptions, and cognition, and participate in the storage and retrieval of memories, right? So we know that this is a very important part of the brain, and it has the most to do with cognition or higher thinking in humans, right? So much like hindbrain, the embryonic forebrain is initially divided along its rostrocaudal axis into transversely organized domains called prosomeres, right? So, like there was a rostrocaudal patterning across the brain, even within the forebrain, there is this patterning that is occurring, and there is division such that different regions are known as prosomeres, right? So just yes. So these different regions that are divided like this are known as prosomeres, right? I forgot to keep a representation or a schematic describing what these prosomeres are, right? But we can read a little about it. So, for instance, prosomeres 1 to 3, they develop into caudal part of the diencephalon, right, from which the thalamus emerges. So we know, we have studied about what is diencephalon. It has three major regions in the mature developed brain. They are the thalamus, epithalamus, and hypothalamus, right? So this is the prosomeres 1 to 3 are responsible for the formation of the caudal part, which is the lower part of this diencephalon, right? Then prosomeres 4 to 6 are giving rise to the rostral diencephalon and the telencephalon. So the upper part of diencephalon as well as the telencephalon, which is a part of the forebrain, that is being formed by prosomeres 4 to 6, right? Then again, the ventral region of the rostral diencephalon gives rise to hypothalamus and basal ganglia, whereas the telencephalon gives rise to neocortex and hippocampus, right? So these are brain areas that we have already understood. So you already gone through. You do not need to remember this. It is just for your understanding that I am telling this, right? Then again, there are some inductive signals over here. So at the beginning of development, we have studied that we thought that the neural tissue development is also going to be responsible due to a result of some inductive signals, that some signal is going and inducing the ectoderm to become a nervous tissue. But over the course, we have already found out that that is not the case, right? Instead, what is happening is that the actual default fate of the ectoderm, which is forming the entire nervous system, is to become a neural cell. And there is some repressive signal that is acting upon it, which is leading at least some cells to go and form the epidermal cells, which will form the skin, right? So it is repressing the natural fate, which is development into neural tissue, and forming the epidermal tissue or the skin, right? But over here in this brain development later on, there are actually some inductive signals that take place, right? Or that perform certain functions. So this inductive signals as well as transcription factor gradients, right? Earlier also we have looked at transcription factor gradients when we were looking at OTX2, right, or the other one downstream at the caudal end, right? So over here again we will look at transcription factors gradients, and we will see how they will establish regional differentiation, right? So even within the forebrain, how regional differentiation is being brought about because of different transcription factors being expressed in different regions, right? So there are two homeodomain transcription factors that are actually present. One is known as PAX6, the other is known as EMX2, right? They are expressed in complementary antero-posterior gradients. Complementary gradients as in that if one is present in the anterior side, the other is naturally present in the posterior side, right? They are not inhibiting one another's occurrence. It is just like that, that one is always found in the posterior side, the other is always found only in the anterior side. uh, in the anterior posterior gradients in the ventricular zone of the developing neocortex. High levels of PAC6 are present at the anterior level and high levels of EMX2 are present at the posterior level, right? The early patterns that are established in part by the local rostral source of FGF signals, which promote PAC6 and repress the EMX2 expression. So, what they are saying is that initially there is some FGF fetal growth factor signaling that is occurring in the rostral region or the front end of the brain, right? Because of this FGF signaling, what happens? It is promoting this PAC6 to be present present as a transcription factor in the front part of this forebrain, and it is inhibiting the expression of EMX2 as a transcription factor in this front part of the forebrain, right? So that is what is happening. This is what happens. FGF subtype is 8. So FGF8 is actually present in this developing neocortex, right? This is a representation of a mammalian brain. Over here, actually, it is a mouse brain, right? So in this mouse brain, you can see these are the olfactory lobes. This is the, cerebrum that is present. Over here is the cerebellum, and this is the brainstem region, right? So this is the anterior end, this is the posterior end. Okay, so over here at the anterior end, you can see that FGF8 signaling is active, right? The FGF8 signaling leads to, this kind of gradient wherein it promotes PAC6 signaling over here, but it represses EMX2 signaling over here, right? This FGF is doing that. So that is why this FGF naturally is showing more of a signal over here in the caudal end of this neocortex, right? So, the spatial distribution of PAC6 and EMX2 helps to establish the initial regional pattern of neocortex, right? So, how is that? Because this end wherein PAC6 is more active develops the motor cortex, the sensory cortex, where EMX2 is more active, it develops the visual cortex and the auditory cortex, right? By default or by nature also, they are not always similar in size, right? They have different kind of distribution depending on what is the area that is, dedicated to that kind of a stimulus or that kind of a function, right? So motor cortex, sensory, somatosensory cortex, you can see are almost equivalent. Visual cortex is a larger area. Auditory cortex again is almost half the visual cortex area, right? But what happens in the EMX2 mutant? So EMX2 is a transcription factor that is active in this lower region, right? If there's a EMX2 mutant, meaning this transcription factor is no longer active over here, right? What happens? You see that there is a shrinkage and of this visual cortex and auditory cortex area. Also, along with shrinkage, you see that they are pushed further down, right? Why is that? Because now the pack signaling that was more active over here, now even if this gradient was a little over here, you can see the gradient is very shallow over here, right? But since this is not present over here, EMX2 is not present over here, even this shallow gradient can induce more form development as what would have happened in the anterior part of the brain, right? So it will be more like the anterior part of the brain because it will be able to manipulate it like that, right? So that is why this motor cortex and somatosensory cortex, you can see, has grown in size like anything, and these two have shrunk in size also, and they have been displaced further down also, right? Similar to this, if there was a PAC6 mutant, right, it would just be a reverse situation. Over there, this EMX2 transcription factor, which is expressing on this end, this EMX2 will have, the effect of the gradient will still function, wherein the PAC, if it would have been present, it would have been overpowered. But now PAC6 is not present, so this gradient will still function, and this visual cortex area you can see has now widened so much. Auditory area has moved further upwards, and the motor cortex has shrunken a great deal, and somatosensory area has also shifted up, right? So these kinds of gradients are actually essential for the proper patterning and proper region-specific differentiation of the brain. Then there are certain afferent inputs also which will contribute to regionalization. Right now, what are these afferent inputs? The nature of the input to different cortical areas influences neural function as well as cytoarchitecture. Right? What's cytoarchitecture? The cell type that is present over there. Right? So what does it mean? Afferent input could be an input from the outside of the body also, right? So we can see that in a normal circumstance, first let us just zoom in the first part of this and study this. So in the normal part of the brain, what you can see is this is a combination of the visual and the auditory inputs that the brain is receiving. Right? Just a moment. Yes. So over here, you can see that there is a visual stimulus that is being received by the brain and an auditory stimulus that is being received by the brain, right? So this visual stimulus is okay. I can't zoom in and use a laser pointer at the same time. Okay, I will just say what I want to say, right? So visual stimulus is coming via retina, auditory stimulus is coming via cochlea inside the inner ear, right? You can see that there are different regions that the stimulus is being, directed to. So this LGN is a nuclei region. Lateral geniculate nuclei, I think, that region wherein this retinal signal is being transferred, and then this region is taking it to the visual cortex area. This visual cortex area is where the higher processing of the signal is taking place, and the brain is trying to figure out what this object is that we are seeing, right? Or trying to recollect maybe if we have already seen that object, if we know what that object is, that kind of processing is happening over here in this visual cortex area. Right, the signal that is coming from here, from the inner ear, is going to the inferior colliculus, right? From this inferior colliculus, it is then going to this MGN, which is another set of nuclei, and then it is being taken to the auditory cortex, right? Where this auditory signal is being made sense of, right? So, the output that our brain's output will kind of look like this, right? So this is what the brain is actually making sense of, that these are the things that are present. It is just a schematic representation showing that this is the output that our brain is giving, right? That maybe this is the gradient of colors that is present or something like that. It is a combination of the two signals, right? If, for instance, this inferior colliculus region that was present, which to which this cochlea was taking the signal, so that it was being carried forward and finally being reported to the auditory cortex, if this particular colliculus region has been ablated or it has been removed, right? So that means the inner ear signal will never be able to, reach the brain, because now the area that was responsible for reading the signal has been removed or has been ablated, right? So what will happen is the retinal signal is only coming, right? It is still going to the LGN, it is still going to the visual cortex area, right? And the visual stimuli we can still understand. Along with this, what happens is sometimes this visual input is getting branched and it is also reaching this MGN region, which was earlier getting signal from the inferior colliculus, right? So earlier this signal used to be the auditory input, right? But since this auditory signal is no longer there, this visual stimuli is branching out and reaching the MGN also, because the neurons are activated in this MGN. Their connections are basically leading to, and they are also activating neurons in this auditory cortex area. But now it will not be, responding to an auditory stimulus, but it will be responding to a visual stimulus. So that is why it has been written as a visually responsive auditory cortex, right? So this is how sometimes, even if there is an accident, even if there's a lobotomy in the brain, if there's a surgery that needs to be done, it is not always that it will impair a person, right? There is some kind of plasticity that is present in your brain, that is some part of the brain can still rewire, and the remaining part can take over the function that was being done by this lost part or lost region, right? So the brain over here has been rewired, and over here you can see that the rewired brain, although the signal looks different, right? You can see the signal is obviously different, but the output that it is giving is exactly the same. So it can still understand, only through the visual signal also, it can understand what was being conveyed, right? So it is a little complex, but what you just need to understand is that there is possibility of rewiring of the brain, right? This is one example of that possibility. It was actually an experiment that was performed in ferrets. So they are other mammals, right? Now, with that, we can end our discussion upon the general development of the brain. I will just go over some topics that I have not touched during this development part. So I have only dealt with development of the brain regions, right? During this embryonic phase, I have not really gone into the actual cell type development that is going on during this embryonic phase. I have not gone into the prenatal as well as postnatal, formation of synapses and innervation. Those kinds of things I have avoided over here, because then it will be a very, very lengthy discussion. I don't think that much is important over here. Maybe if we take another course which has just to do with the development of the nervous system, maybe we can talk about those things in detail over there, right? But this was one part that I did not really want to skip. It is actually a very interesting part, that our brain also shows sexual dimorphism. Right. So there are some mechanisms obviously that are at play due to which this is happening, right? So there is some sexual differentiation of nervous system also that takes place early on, due to which there is this sexual dimorphic pattern that is existent in the brain. Now, what is sexually dimorphic? It means that it will differ between a male and a female, right? Or it will differ between the two sexes of that species, right? So let us go through certain examples or illustrations as to how there is this sexual dimorphism that is present in the nervous system, right? So this represents one of the mechanisms due to which this sexual dimorphism is there, and that is basically due to the presence of steroid hormone receptors, right? So we know that the sex hormones that are present in our body, which are either the estrogen or the testosterone, mostly, right? They are both steroid hormones, right? Steroid hormones means there's a cholesterol backbone, and on that there are different kinds of functional groups that are added, due to which either it will become an estrogen, estradiol, etc., or it will become a testosterone molecule, right? So these are all steroid hormones, right, that are present in the male as well as in the female. So because these steroid hormones are there or these molecules are there, there are also some steroid hormone receptors that are present in our body, right? Now this steroid hormone receptor has this basic structure, right? So this is the canonical receptor for steroid hormones. That is, canonical means the general basic receptor for steroid hormone, and these are ligand activated transcription factors, right? What does this mean that it is ligand activated? That it has this N terminal domain, right? Yeah, this is the structure. Like, what does ligand activated means? That if this hormone comes into contact with this receptor, it will basically activate the receptor for that particular hormone, and then the signaling will occur, right? So what is the structure? It has an N-terminal domain, it has a DNA binding domain, and it has a ligand binding domain, right? So this ligand binding domain will bind to the ligand or bind to the hormone, right? DNA binding domain is going to go and activate whatever transcription factor needs to be activated in the DNA inside the nucleus, right? So, this is how they act, right? So you can see that where are these steroid hormones coming from? They are basically being secreted by our gonads, right? So gonads meaning ovaries or testes, right? So they are secreting these steroid hormones, and these steroid hormones, they are in the bloodstream, they are going to different organs of our body, they are also reaching the brain, right? So how do they enter the brain? These sex steroid hormones are hydrophobic, right? That is, they are not, they avoid water, right? And they enter the circulation by diffusing across the plasma membrane of the steroidogenic cells in the gonads, right? This is where they are being released. After that, what is happening? They enter the target cells in distant tissues, because now they are in the blood, they can travel the entire body. So even distant tissues such as brains, they enter the target cell by passing through the plasma membrane and bind their cognate receptors, right? So what they do is, steroid hormone receptors are generally present within the cell, right? So because these steroid hormones are they are hydrophobic, and they are also they have this cholesterol backbone, right? So it is feasible for them to pass the plasma membrane, so that they can do, and inside the cell they will bind to their receptor, right? So these receptors are generally not on the surface of the cell, they are inside the cell, right? The steroid hormone receptors typically exist in a multi-protein complex. You can see that there are multiple proteins that are there. It is not just this receptor, there is this chaperone protein also along with this receptor that is present. With chaperone proteins in the cytoplasm of the hormone responsive cells, right? Then the ligand binding, that is when this hormone is going and binding to this hormone receptor chaperone complex, this promotes dissociation of the receptor from the chaperone complex and translocation into nucleus, right? So once this binding occurs, what happens? Chaperone protein is left behind, this receptor is dissociated, and this receptor along with the ligand, it goes and it binds, goes into the nucleus, binds to the DNA that is present in the, or chromatin that is present in the nucleus, right? In the nucleus, the receptor is thought to bind to a hormone response element as homodimer to modulate transcription of target gene. And generally, this one receptor ligand complex will not bind to this particular DNA alone. It will generally have to be in a homodimeric form, that is, two of the same hormone and hormone receptor complexes. They come together, form a dimer, and then they activate this pathway of transcription, right? So this is a summary of that mechanism, right? So that is how it generally works across species, right? Over here is one of the examples of why there has to be a sexual dimorphic pattern in the in organisms, right? And in this particular case, how does that happen, right? So over here you can see this one Drosophila. These are Drosophila basically, or the fruit fly. And over here we are looking at the control of male courtship in the fruit fly, right? So over here, this one fly that is marked with an asterisk is actually the male, right? Now, what does this male do? The male has to basically, the male is the inferior species over here in Drosophila or fruit fly, and it has to convince the female to basically let it, uh, let it mate with the female, right? Over here you can see that there is a pattern of this male courtship that is there, right? It begins with orienting. First the male will orient itself towards the female, right? For that it will obviously need to use its senses, use its antenna, use its sense of smell, if there is any sense of vision, if there is any, right? So that it can orient itself to the female. Then what it will do, it will go and it will tap the female, right? So it will continuously go and tap the female, right? Then it will also have some wing vibration, right? It will shutter its wing very quickly so that it can draw attention from the female, right? Then it will lick the posterior of the female and it will attempt copulation, right? If this female is fine with that, it will stay. Otherwise, this female will, just get rid of this male fruit fly. That is what happens, right? Now, during this courtship, the male is doing all this, but it is basically being controlled by the brain, right? Obviously, there are sex hormones that are responsible for this, but the hormones also have to stimulate some system in the brain so that the body knows what to do, right? So that is what is happening over here, and that is what is making this male different from this female, right? The hormone that is being secreted and the kind of effect that hormone is happening is having on the brain, right? So this is what happens. This is a male brain and this is the female brain, right? So in the male brain, you can see these two white circles. They basically represent the ommatidia, or the eyes of this compound eyes of this fruit fly. Over here, you can see that this particular region is where this particular transcription factor, this green label is for a transcription factor, which is known as fruitless, right? Which is responsible basically for this mating behavior. We will read about it further, right? Over here, you can see that in male, over here in the ommatidia, it is active, while in female brain, it is missing, right? So that is why this kind of behavior is being shown by the male and not the female, right? And this might be helping it orient itself to the female in the first place, right? Or look at it, where to go and touch the female, how to where the wing stutter should happen. All these signals might be coming from this stimulus over here, right? Then again, in this yellow circles, you can see this region, which is the antennal region. Over here, you can see this is highlighted in this yellow circle, right? Over here also you see a difference in pattern between the male brain and the female brain, wherein it is highly activated and expressed over here in this antennal lobe region in the male, but not that active in the female brain, right? So these are some differential patterns that are present in the brain of this fly, right? So all steps in male mating ritual require expression of a sex-specific splice variant of the fruitless gene, which is known as FRU, right? So the FRU gene encodes a male-specific splice variant that is necessary and sufficient to drive most steps in a male fly courtship ritual, right? The spinal nucleus, okay, so that is the end of that part, right? Now, there is another kind of sexual dimorphism that we can study about, which is there in case of mammals, right? It is there in humans also, but over here, the study that we are referring to has done it in case of mice, right? So, where is the sexual dimorphism occurring? It is occurring in the spinal nucleus of bulbocavernous muscle, right? What is spinal nucleus? It is a region where the cell bodies of the neurons are present, which are going and innervating this bulbocavernous muscle, cavernosus muscle, right? Now, what you need to understand first is that this particular spinal nucleus is found in the male lumbar region, but is greatly reduced in the female. Right. So this region itself, where the cell bodies of the neurons that are going and innervating this region, is highly developed in the male lumbar region, but it is almost absent in the female lumbar region. Right. Now let us first go through this representation, right? I will just zoom into it. Over here you can see a mouse, right? Then at some point in the lumbar region, you see that there is this region that is occurring, which has this SNB motor neuron, right? The cavernosus motor neuron, which is going and it is innervating the cavernosus muscle, right? So what you can see over here, this muscle basically has an androgen receptor, right? Androgen is a male specific hormone, that is testosterone is a kind of androgen hormone, right? So this in during development it is present, this bulbocavernosus muscle is present both in male and female, right? Over here, however, you can see that because there is an androgen receptor that is present in the male, and this receptor can be stimulated by the testosterone, which is present in the male. That is why it comes and binds, and this binding releases some factors, which are the trophic factors, which lead to basically the innervation of this muscle via these neurons that are present in the spinal nuclei region, right? However, because these trophic factors are not present in the female, because this binding of androgen receptor and androgen is not occurring over here, because of lack of testosterone, what happens is the innervation is not happening. And since the innervation is not happening, the important thing to know over here is that if a neuron has not innervated, or it does not have any function, it will die basically, right? So this region will not be developed over here in the spinal nucleus region, corresponding to the male region in the lumbar section of the female, right? And this will subsequently lead to muscle atrophy and motor neuron death, right? So muscle will die, and corresponding motor neuron will also die. But over here, because there is a viable connection that is there between this motor neuron and the muscle, that is why the muscle will be viable and motor neuron survival will still happen, right? So now this is a muscle that is only developed in the male, and it is not being developed in the female, right? So it will obviously have something to do with the some sexual characteristic that is present only in the male and not in the female, right? So this muscle actually is responsible for the copulation. I think it is responsible for the ejaculation part of this copulation that is there in the higher mammals, right? So that is a function that is only required by males and not females. That is why the sexual dimorphism actually occurs, right? So the motor neurons of the nucleus are present in both sexes at the birth, but lack of circulating testosterone in the female leads to death of the SNB neurons and their target muscles, right? Over here, again, this is a representation from the mammal, wherein this is the dendritic spines, right? Dendritic spines are basically the neuronal cells. This purple is neuronal cell bodies, and these are dendritic spines, or the dendrites that are going to this region. Over here, in the intact male, you can see that dendritic branches of SNB neurons is regulated by circulating testosterone in the adult male rats. Uh, in the males, the dendrites arborize extensively within the spinal cord, as is seen over here. But in fact, if the male mouse was castrated, so in castration, what happens is the circulated testosterone level also goes down, because of which fact that arbors are the fact that the arbors are pruned in adult castrated male rats, which is there in the lower photo, the evidence is that dendritic branching depends upon androgens, right? Also, another reason is because that muscle is no longer there, so the corresponding motor neuron has no function now, so it has also been lost, right? Then this also exists in birds, right? So there is a sexual dimorphic pattern that exists in avian songbirds, in the avian song circuit. So in the songbirds, they have a dedicated neural circuit for song production and learning with distinct components which are contributing to learning or production of songs, right? This is what it looks like, right? Many of these components are sexually dimorphic in songbirds with in which only one sex sings, right? So, like there are certain birds in which only one bird will sing, only one sex will sing, the other will not sing, right? Or for instance, in a peacock, right? Only the peacock will dance, the peahens will not particularly dance, right? So that kind of a thing happens in songbirds also. Over here, you can see that in the male, the axons of the HVC neurons, which is a neuron that is originating over here, terminate on the neurons in the RA nucleus. So this is another set of neurons, and the axons of HVC are actually terminating in the nucleus of this RA, right? Whereas in the female, the axons terminate in a zone surrounding the nucleus. It is not entering the RNA nucleus, it is only in the zone surrounding the nucleus. So just by this, what can you guess that in the songbird, the males are actually the ones that might be responsible for the production of the song, right? Not the females, maybe, right? The sexual dimorphism in cell number and connectivity of these regions is regulated by estrogen. Over here, it is being regulated by estrogen because only the female has estrogen. That is why it is showing a different kind of pattern. And because over here in male estrogen is not present, it is showing a different kind of pattern, right? Sorry. Then this kind of a dimorphism also occurs in the human brain. Over here, you can see there are labeled four regions in the human brain. It is in the interstitial nucleus of the anterior hypothalamus. So INAH, right? So there are these four regions that are labeled. This this is a hypothalamic region. E is a cross section from this region and P is a cross section from this region, right? So while you can see that INAH 1, 2, 3, 1, 2, and 4, right? So 1, 2, and 4 you can see in male. This is present in male, present in male, present in female, right? Similarly, 3 and 4 are also present in male, but 3 is almost absent in male. It is only present in almost absent in female, only present in male. 4 again is present in both, right? So you can see that the INES3 is significantly larger in men, right? Uh, okay, so I have not described this further, but again, this might be responsible, this might be a result of some of the steroid hormone induced characteristic, right? Also, this kind of sexual dimorphism is widespread in the adult human brain, right? This is a representation of a MRI image, which has basically studied and which study the measured the volume of many brain regions in adult men and women, right? So, whether like they had multiple, subjects. In these multiple subjects, they have done the MRI, and based on the MRI, they have seen that, okay, is it true that the forebrain is more enlarged in the females, or more enlarged in the males? Is the visual area more active in the female, or is it more active in the male? Is the motor cortex region more active in the more widespread, or it covers more portion in the male or the female? And similarly for other regions, right? So over here, this reddish peach color, it basically signifies area that is proportionally larger in the female brain, right? So it includes some of the this cognitive areas such as somatosensory and motor sensory regions. Hippocampus, it includes, right? The C, we can see some region of this occipital lobe, it is including some of the other regions also. And there are some regions that are proportionally larger in the male brain, which are represented in this blue color, right? So that is also there, that uh, this kind of sexual dimorphism in the brain is also seen in humans, right? So that brings us almost to the end of this discussion, right? So the volume of each region was actually normalized to the size of the cerebrum for both sexes. So yeah, that is how most of the calculation is done in biology. So we'll not go into detail of that. Sex differences were significant in many regions, including several cortical areas that likely mediate cognitive functions, right? Over here is another example of this kind of sexual dimorphism that is seen in the brain, human brain, and how it may correlate with gender identity and sexual orientation, right? So, for instance, now we are aware and we are getting educated more and more that it is not necessary that the human body is as simple as either male or female. There are some variations also that occur in nature, due to which there could be a homosexual men or homosexual women that are there, right? So there are some sexually dimorphic patterns of olfactory activation in the brain, which correlate with the sexual orientation also of a person, right? For instance, over here you can see that this is a response to some kind of stimulus, which is again an olfactory stimulus, and you can see that the response is very similar in homosexual men and heterosexual women, right? It is almost stimulating the same area, right? In this case. Or what is another similar response? Similar in this left amygdala region connectivity, also you can see it is very similar between heterosexual women and homosexual men, versus it is very similar in heterosexual men and homosexual women also, right? You can see. So that shows us that our sexual orientation is basically going to affect the kind of brain dimorphism also that is there within us, right? Yeah, so that was actually the final slide. That brings us to the end of this discussion. As I said, I will end it with the discussion of with the completion of this development part of the brain. I am aware that as per my scheduled lectures, there's just one that is remaining, which we'll do on Sunday. But there are three topics that are remaining: the sense organs, the memory, and the diseases. So I will actually pick up one of the topics and we can deal with that in the next session, right? And the rest of it, I will try to form a separate course out of it, so that we can, because development itself took some time, right? So I think we can split it into two classes each one of that. So for that, we will maybe even if I ask for extension of this course, it will be extended by almost as many classes as we have already done, right? So we'll probably not do that. Maybe we'll, come with another subsequent follow-up course for this one, right? So that is what we are planning to do. Now that we are seeing that it is so time taking to talk about or discuss these things, let me just stop sharing. Yeah. So with that, is there any query at all? So if there is, you can obviously turn on your mics, you can speak up. Or you can also reach out to me in the discussion forum of the Google group that we have. This week I have been a little busy. I have not really checked the group, but you guys have not been very active over there also. So I don't think I might have missed anything, but I'll check nonetheless. Okay, so I'll also end the recording. Thank you everybody for attending.